Alrighty guys, welcome to my video. Today we'll be talking about DNA and the general knowledge required for year 12, the term 1 biology exam for the 2022, end of 2022, beginning of 2023. Anyhow, these are some notes about the total, what we need to know for all of term 1 for biology, but right now we're just going to be looking at the general DNA information that we need. So what we have to begin with is obviously, what is DNA? DNA is made of repeating nucleotides wound into a double helix. The sequence of bases codes for the RNA and the amino acids that form polypeptide chains. Now polypeptide chains we'll be looking at the end, towards the end of this video or the end of this series. And DNA is made up of repeating nucleotides. Nucleotides are phosphate group, deoxyribose sugar, and nitrogenous base, as we can see here, and sequence of bases codes for the RNA, even though they only have 4 bases, which is A, T, G, C, which is also known as adenine, thymine, guanine, and cytosine. We're still able to see that they're able to code for thousands of different polypeptide chains, or also known as proteins, inside the body. And we'll be able to learn more into that. Now DNA, it can be found in the nucleus, mitochondria, and chloroplast in eukaryotes. In prokaryotes, it's only able to be found in the nucleus. Now, what's it called when it's not in the nucleus? It's called a non-nuclear DNA. Pretty easy. Nucleus, non-nuclear. But the reasoning for this is because they're in the cytoplasm. Alright? Now, we're going to move on. Base pairing is when you have the A with the T bond and the G with the C bond. They bond, adenine and thymine bond with a double hydrogen bond, and guanine and cytosine bond with a triple hydrogen bond. Now, how does this happen? The adenine is a double-ringed purine base, and the thymine is a single-ringed pyrimidine base. That also applies for G and C respectively. And what we can learn from this is that adenine, which is a double-ringed purine base, can only bond with the thymine, which is the single-ringed purine base, and G can only bond with C. This is so that the DNA backbones are equidistant from each other, which means that they remain the same distance away from each other. This is obviously important. They bonded 3 hydrogen bonds. AT has a weak double bond, and GC has a weak triple bond. Now, we need to move on from this. How was DNA and the double helix structure discovered? We give credit to this to James Watson and Francis Crick. They're known as the people who found out the double helix structure. How did they find it? Let's find out. So Watson and Crick worked together. Watson was the guy that looked at the sketches and he understood what they were supposed to look like, the double helical nature. And Crick was the mathematical guy who looked at the x-rays and found out his mathematical methods. Franklin was a woman who took an x-ray of the DNA, but during her time, women weren't really appreciated. So Wilkins stole the x-ray and showed it to Watson and Crick. Now, they were able to use all this to establish the double helical nature of the molecule. Here we can see a more better explained version of what I just said. Now, DNA has all requirements expected of hereditary material, all requirements expected of something that can be taken from your parents. Now, it can be coded in coded form through the 4 nitrogen bases, and this allows it to be able to have the instructions for the formation and functioning of cells, even though it only has 4 nitrogen spaces. This is important. There's many formations and many functions, and it can do this all with 4 nitrogen spaces. We'll look into how it does that through different combinations. Now, it has a structure that allows for replication because it's a double-stranded structure, and if you separate the double strands, you're able to have 2 strands that can be replicated to produce the same exact strand. Now, this means that it can be transferred from one generation to another through chromosomes and being carried by gametes. Now, this is meiosis, and the replication is usually done in mitosis. Now, we have a DNA model, DNA kit, and we'll be able to talk about the limitations and why we should use the DNA model and such and such. Now, the DNA model kit is good because it allows us to see and understand the double helix structure of the DNA, which is usually not visible to the naked eye, or which is not at all visible to the naked eye. But what we're going to write in the exam is that it's not visible to the naked eye. And it is easier to understand the structure of DNA, including the nucleotides, and you need to expand on this. The nucleotides phosphate sugar base. Make sure you make sure that you say it's the deoxyribose sugar, phosphate group, and nitrogenous bases during your exam. Make sure you be specific. And also the base pairings, obviously the hydrogen bonds in between them. This is good because we're able to understand the DNA better, and we are able to visualize it. And also this is obviously the better, and also it might be having some limitations, such as it being oversimplified, as it does not show the actual size of nucleotides, which is obviously minuscule. And there's obviously thousands of nucleotides in a one DNA strand, and that's in one gene, honestly. And there's like hundreds of genes or thousands of genes in one DNA strand. And it also doesn't show the hydrogen bonding between bases in the DNA model. Now, when we have the DNA replication, we're able to show the hydrogen bases. DNA replication will be talked about in the next video, but it's here if you guys want some notes on that. Thanks for watching.